Welcome back to Vaccine Design course. Now, under the functional analysis of antigenic protein structure, our next step would be to check for the T cell epitopes immunogenicity prediction. Okay, so now what this tool uses is amino acid properties as well as their position within the peptide to predict the immunogenicity of a peptide MHC complex or a PMHC complex. Now let's start with the task. Let's go to this particular link. Okay, so what you need to do is go for IEDB T cell epitope prediction tools. Now under that you have the T cell epitopes immunogenicity prediction, which is what we are going to do. We completed the MHC binding prediction, processing prediction. Now it's the immunogenicity. Okay, so click the first link, which is T cell class 1 PMHC or the peptide MHC immunogenicity predictor. Okay, so this is the home page of it. As you can see, you need to now you can always let the help section be open and just keep it aside. Now you need to enter your peptide sequence. So Control A, Control C and Control V. Okay, so here you need to remove your FASTA header or else it won't be detected. Okay, just paste your plain amino acid sequence and then specify which positions to mask. So here in the help section you can see the masking option. What it does is it will mask the designated peptides from the immunogenicity score. So now read the whole thing, but so it says about the HLA molecules, right? HLA, which is nothing but the human leukocyte antigens, which are the genes in the major histocompatibility complex or the MHC that help code for proteins which differentiate between the self and the non-self. So this the HLA play a significant role in disease and immune defense. Okay, and they are beneficial to the immune system, but they also can have detrimental effects as well. Okay, so by default this positions to be masked is the first, second and C terminus amino acids, or you can custom design it as well. Okay, so let's make it default and click the submit button. So as you can see in the results, the MHC class 1 immunogenic peptides that are present in the protein. So here each row in the table corresponds to the prediction for an individual peptide. So here the table consists of peptide, length and score columns, right? So by default the prediction results are sorted by the descending score values. So it's descending score values. However, the table can also be sorted by clicking on the individual column headers like this. Okay, so higher the score or the higher score indicates a greater probability of eliciting the immune response. So this is actually the highest score, right? Minus. So since it's minus, so this particular peptide has a greater probability of eliciting an immune response. Now, so that's how this one does. Okay, so next what we'll be doing is to see how much immunogenic are the T cell peptides. You need to go to the third link of the or under the T cell epitopes. Now again third link, which is CD4 T cell immunogenicity prediction. Okay, so let's go to the third link. So this is the home page of it. So it is CD4 T cell immunogenicity prediction. You need to paste your sequence, sort the peptides by percentile rank. So it's again by position. What we need is percentile rank. The prediction method, let it be IDEB recommended. Now you can go to the help section and see what it says. You can select the maximum combined score threshold. You can select it. So let's make it by default, which is 50, and then go to the submit button. Okay, now here when you see the results, it's the immunogenicity score which is important to notice. Okay, let's wait for the results. By the time we'll go to the help button where it says the step-by-step procedure, which we already did. Okay, so it says about how to sort or how to choose the sorting function, how to choose the threshold and everything. Now the results are also interpreted here, but let's go to the results section. Yeah, so here you can see various sections in the column. Like I said, it's the immunogenicity score which is really important. Okay, which we have here: 73, 65, 89, 84 and 81. So now you can see various other scores along with the protein number, protein description, peptide, start and end. You have the combined score, which is, it's from a median percentile rank of 7 alleles and immunogenicity. That's the combined score. Okay. Now you have the peptide score, which says the 9 amino acid peptide score that is used by the immunogenicity neural network model. Then you have the median percentile rank, right? That is, it's the median percentile rank of 7 alleles and the individual percentile rank. Okay. Then you have the individual percentile ranks, which 7 alleles. So yeah, so you can go to the help button and see what all it means: the protein number, protein description, and about the median percentile rank, and about the 7 alleles, right? But the immunogenicity score is what is important for us. Okay. If you click here, you can make it arranged, right? So make it the highest number. Highest immunogenicity score means that much immunogenic the peptide is. Okay. Now, so this is how you need to interpret your result. Now, when you go back to the tools IEDB page, or let me just go back to the PowerPoint. Okay. So we finished the T cell epitope prediction. Now let's see the population coverage, or let's see in how many people this particular protein sequence is present. So for that, again, you need to go to the main site of IEDB. So this is the home page of IEDB. Go to the epitope analysis tools. Under that, click on population coverage. Okay, remember this is done based on MHC alleles, that is only for T cells. So let's give the number of epitopes. You can give it, let's make it as 50. Okay. Then you have something called as query by area, country and ethnicity. Make it so let's select India, and then you can select the calculation options. Either you can select for class 1 MHC, class 2 MHC. So if you select only this, it's just the class 1 MHC separate. If you want both these combined, you will get the combined values as well. So for the demo purpose, let me just show you how it is shown when class 1 is selected, class 2 is selected, and class 1 and 2 combined is selected. Okay. So let's select all these three. Now when you scroll down, you have something called as epitopes and MHC restriction alleles, which you have to write it here. What you have to do is, remember that we have done the MHC1 prediction server from IEDB and we got the results, right? So you need to copy and paste the results here, which means the epitopes, you need to write the peptide which was shown in the result. So this was the MHC1 result. You need to write the peptide there in the epitope section, and in the MHC restricted alleles you need to write your alleles. Okay, that's how you need to do. So since we have given the class 1, class 2 and class 2, you need to write the class 1 MHC result and class 2 MHC result as well. Okay, so here let me just copy the peptide here, control copy, and I'm pasting it here. So this is the MHC1 result. I'm copying the allele and then pasting it here. Okay, how much ever peptides that you need, you can. Okay, so again copying the second epitope and the second allele. Okay, so for now I'm copying 2 peptides from the MHC results, 2 peptides and 2 alleles, and I'm copying the 2 peptides and 2 alleles from the MHC2 result as well. Okay, so what I did is, the first 2 alleles and first 2 epitopes seen are from the MHC1 result, and the other 2 are from the MHC2 result. You can either save it and browse it from your PC and paste it here. Okay, anything. So after doing that, you need to click the submit button and then wait for the results. So here you can see the table view, and then there is a graphical representation also. So all these are for class 1. So this is how the results are interpreted when class 1 class 2 and class combined are selected. Okay, that is, the coverage is mentioned, the average hit, as well as the standard deviation is also mentioned here. So the class 1, when it goes, how it looks like. Now this is the graph, okay, where you can see the MHC class 1 about the coverage, average hit, and the PC90, and then about the MHC class 2, the representation of it, and the combined representation is also here, MHC class. Okay, take the help and see what population coverage is and how the output is analyzed. It's the same thing that we did. Okay, which says, so here when you give more alleles, you will be getting a graph like this. Okay, so in the graph, the x-axis represents the number of epitope hits. So these were the epitope hits, and the y-axis represents the percentage of individuals as well as the cumulative percentage of population coverage. Coming back to the results, in the graph, if you want to view it in a table format, you can select the view chart data, which represents the in the tabular format. So here, as you can see, the number of epitope hits, HLA combination recognized, 0. So in 0, as you can see, it's the percentage of individuals is 94.13, and the cumulative percentage was 100, right? And can you see the first and second? That is the number of epitope hits. So when it's the number of epitope hit is 1, the percentage of individual is again 0, but the cumulative percentage is 5.87, which means it's the combined one. When there are 2 epitope hits or the HLA combinations, the percentage of individuals, there are 2 alleles recognized, the percentage is 5.87. Okay, so this is for MHC class 1. Now let's look at the combined version. Let's look at the tabular format. Okay, so it's pretty much okay. When it's combined, there are 1, 2, 3, 4 epitopes, but for the 2 epitopes, it's the percentage of individuals is more, it's 6.41 when it is compared with the percentage 0, which is again the number of hits, which is 0, the percentage of individuals is 93.55. okay, now let's see the coverage of individual epitope in India. Okay, so when you click here, it says about the epitope 1, 2, 3 and 4, the 4 epitopes that you have given, the coverage that you see in India, that and the total HLA hits. Okay, so you also can see the restricted and the non-restricted ones as well. Okay, so this is how you need to interpret to see how many people, or to see in how many people this particular epitopes are present. Okay, so in total, how much epitopes that we have given is, we have given 4 epitopes. That's what was seen in the combined version, right? Yeah, so 4 epitopes were given, where 2 epitopes were MHC class 1. So that's why in the class 1 there is just 0, 1 and 2, whereas in the class 2 again 0, 1 and 2, because only 2. But when it's combined, in total there are 4 epitopes. So out of it, how much percentage of individuals, in how many epitopes are present in how much percentage of individuals, that's how you need to interpret. Hope this is clear. So this is about the population coverage. So by this lecture, we have come to an end of functional analysis of antigenic protein structure. The next step that we'll be following is, again, we'll see in the next lecture. Okay, thank you.